The actual form of the multiple topographic maps that are laid across the cerebellar cortex is illustrated in this picture. And again, you can see that in the vermis portion of the cerebellar cortex, that is the region of the cerebellum that's controlling axial or trunk musculature and proximal limb musculature. Then when we move into the intermediate or paravermal hemisphere, that region of the cerebellar cortex is controlling distal musculature, and then the lateral aspect of each hemisphere is again mainly involved in motor planning. Next point we can make is kind of an overall functional view of various parts of the hemisphere. Because the vermis and the intermediate zones have functional topographic maps of all the skeletal muscles of the body, we basically say that the vermis and the intermediate zones of the cerebellum are involved in ongoing control of motor execution. In other words, they're promoting the smooth execution or of any contracting muscle, regardless of whether it's axial or limb located. And one of the things that's unique about the cerebellum is that it is getting much of its input, or a large component of its afferent input, from the contracting muscles themselves by way of proprioceptive input generated mainly by muscle spindles and Golgi tendon organs that enters basically in the dorsal roots of all spinal nerves and finds its way up to the vermis and intermediate zones. As we said earlier, the lateral aspect of the cerebellar hemisphere is mainly involved in motor planning. Much of its input is coming directly from the motor cortex itself. There's basically a plan of the intended movement being sent into the lateral hemisphere, probably indirectly by way of axons of the corticospinal tract. And in addition to getting cortical input about the intended movement generated by cortical neurons, note that the inferior olivary nucleus, which we used as a major structure to help us identify medulla brainstem sections, plays a major role in cerebellar function by acting as an error detector. Basically, its axons project into the cerebellum and influence cerebellar cortical neurons, which we'll see in a moment, to change how a movement is performed basically in the future. Finally, the flocculonodular lobe is getting much of its input from the vestibular system, and as such, that's logical because its major role is to help maintain upright posture and balance and also promote the smooth execution of conjugate gaze. Next point that's made at the bottom of the slide is how does input get into and get out of the cerebellum? Input and output of the cerebellum utilizes three pairs of cerebellar peduncles. There are a pair of inferior cerebellar peduncles that mainly arise from the medulla, carrying input into the cerebellum from the spinal cord and the olive. The middle cerebellar peduncle is relaying information from motor cortex into the cerebellum, so both of those are major afferent pathways. The major way out of the cerebellum basically is the superior cerebellar peduncle. So basically we say that there are two ways in and one way out in terms of cerebellar generated information. Next slide illustrates the cellular organization of the entire cerebellar cortex. Cerebellar cortex is organized into three layers. The outer layer of the cerebellar cortex is largely acellular, populated mainly by parallel fibers, which are axons of granule cells, and it's known as the molecular layer. The Purkinje cell layer is the middle layer of the cerebellar cortex, and arguably it's the most important layer of the cortex because it contains the largest neurons in the CNS. These are the Purkinje cells. But it's the most important layer of the cerebellar cortex because it is only the axons of those Purkinje cells in that middle layer which will actually leave the cerebellar cortex. Finally, the deepest layer of the cerebellar cortex is the granule cell layer. And granule cells are intrinsic neurons of the cerebellar cortex whose major goal is to synapse with Purkinje cells, but their other claim to fame, as we'll see, is they are the only excitatory neuron within the cerebellar cortex. Every other neuronal cell type, including the Purkinje cells, are inhibitory. The next point we want to make is what is the nature of all of the afferent input to the cerebellar cortex? As we just mentioned, all of the neurons in the cerebellar cortex are inhibitory except for the granule cells. But in contrast, all of the inputs to the cerebellar cortex are excitatory, and they come in two forms. They are entering in the form of excitatory mossy fibers, or they're entering in the form of what are known as excitatory climbing fibers. Climbing fibers come from one location. They come from the inferior olivary nucleus, which was a dominant feature of our upper medulla sections. And as such, they are providing an error detection mechanism to tell the cerebellum that it has to adjust its motor control so a motor task can be performed smoother the next time. Mossy fibers constitute all other sources of input. And as we indicated, the other major sources of input are coming indirectly from motor cortex, they're coming indirectly from the spinal cord, and they're also coming in from the vestibular system. All of these sources of input enter the cerebellum in the form of mossy fibers. And again, since they are excitatory, all of these inputs are probably utilizing glutamate as their neurotransmitter. Glutamate is probably used by more than half the neurons in the entire central nervous system. Next, let's look at the organization of the cytology and wiring that makes up the cerebellar cortex. As we already indicated, there are three cell layers, or three layers that make up the cortex: an outer molecular, a middle Purkinje cell layer, and an innermost granule cell layer. And first point we want to make is that the goal of all the excitatory inputs, the two classes of them, either climbing fibers or mossy fibers, their goal, once they reach the cerebellar cortex, is to influence Purkinje cells. Because Purkinje cells, as indicated here, are the only neuron whose axon leaves the cerebellar cortex. And once it leaves the cerebellar cortex, the Purkinje cell axon synapse on deep cerebellar nuclei that are situated down in what we call the medulla of the cerebellum. So here's the differences. All of the two sources of excitatory input, climbing fibers and mossy fibers, we said are excitatory. And on their way to the cerebellar cortex, both of them send excitatory collaterals to modulate the activity of the deep cerebellar nuclei, in essence providing those deep cerebellar nuclear neurons with input from what's going on with a motor plan or feedback from a series of skeletal muscle contractions. So the goal of the inputs once they reach the cortex is to influence the firing of Purkinje cells. Climbing fibers are fortunate in that they provide a direct, powerful, monosynaptic input to Purkinje cell dendrites. Mossy fibers, on the other hand, yes, they are excitatory, but they only have an indirect influence on Purkinje cells, because all mossy fibers, regardless of their source of input from spinal cord, indirectly from motor cortex through the pontine nuclei, or the vestibular nuclei, all of the mossy fibers enter the cerebellar cortex and synapse on granule cells. But then granule cells send their axons up to the molecular layer where they bifurcate, forming parallel fibers that then go over and synapse and provide an additional excitatory input on Purkinje cell dendrites. So, the take-home message so far is all of the inputs are excitatory by way of climbing fibers and mossy fibers. All of the output of the cerebellar cortex is inhibitory and is mediated exclusively by Purkinje cell axons. The next slide summarizes the neurotransmitters used by cerebellar cortex neurons. As we said previously, the only excitatory neuron in the cerebellar cortex is a granule cell, and it, like both sources of input, utilizes glutamate as its excitatory neurotransmitter. All of the other neurons in the cerebellar cortex are inhibitory, and they all utilize GABA, gamma-aminobutyric acid, as their inhibitory neurotransmitter, including certainly the Purkinje cells, which, as we said, are the only axons that leave cerebellar cortex on their way down to the medulla of the cerebellum to synapse on deep cerebellar nuclei. Next is, where do and what are the names of the deep cerebellar nuclei that are receiving a synapse from a Purkinje cell? Well, we know that just as the Purkinje cells are situated topographically throughout various parts of the cerebellar cortex, we have vermal Purkinje cells, paravermal ones, and lateral hemisphere ones. Those Purkinje cells are topographically linked to a specific deep cerebellar nucleus, as illustrated on this picture. And the point we want to make is the deep cerebellar nuclei are known as the fastigial nucleus. The interposed nuclei are actually two nuclei that are linked together, known as the umbelliform and globose. And the largest and arguably one of the most important cerebellar deep nuclei is the dentate nucleus. The point that's made on this picture is we can see that various parts of the cerebellar cortex are topographically linked by their Purkinje cells to a specific deep cerebellar nucleus. The fastigial nucleus is receiving Purkinje cell axons from the vermis region of the cortex. The interposed nuclei are receiving Purkinje cell axons from the paravermal or intermediate hemisphere, whereas the dentate nucleus is being driven by Purkinje cell axons emanating from the lateral aspect of the cerebellar hemisphere.